The overall goal of the following experiment is to observe the effect of Acf1 or SNF2h knockdown on E4orf4-induced cell death. This is achieved by generation of cell lines in which Acf1 or SNF2h shRNA expression is conditionally activated by addition of doxycycline, resulting in reduced levels of Acf1 or SNF2h protein. As a second step, cells of the cell lines that have been obtained are treated with doxycycline to reduce Acf1 or SNF2h expression, or are left untreated. Next, E4orf4 is expressed in the cells with or without shRNA-resistant Acf1 or SNF2h, in order to investigate the effect of Acf1 or SNF2h on E4orf4-induced cell death. Results are obtained that show the impact of Acf1 or SNF2h levels on E4orf4 toxicity based on counting of nuclei with apoptotic morphology using the DAPI assay.
The main advantage of this technique over other methods, such as transient transfection of siRNAs, is that all cells express the shRNA and the percentage of cells co-expressing it together with additional plasmids is higher.
This method provides insight into the mechanisms underlying E4orf4-induced cell death, but it can also be applied to the study of other pro-apoptotic proteins. In addition to Anna, Rakefet, a researcher from my laboratory, will be demonstrating parts of this procedure.
To begin the procedure for generating inducible cell lines, plate T-Rex 293 cells at a density of